Good morning, everyone. This is the second session of the bioinformatics course, and we're going to continue what we started last time. So, last time we discussed the central dogma of molecular biology, and we highlighted the fact that major bioinformatic databases are shaped and interconnected accordingly. We mentioned that in addition to major databases which index DNA, RNA, or protein sequences, we also have the so-called secondary databases that contain information about polymorphisms, about homologies between different species, epigenetic alterations. For the RNA, we have expression of the RNA molecules, non-coding RNAs, ESTs, as well as protein domains and interactions. So today we want to discuss NCBI Gene database, which has a central position among databases. But before doing that, let's go and take a look at the list of the databases to see if we can identify some of the databases that I mentioned in the previous slide. So here we have nucleotides, we have protein, and we have structure. So these are sort of primary databases that index information about nucleic acid sequences and protein sequences and also 3D structures. So we have three databases which contain information about intra-species differences or polymorphisms. We have SNP in here, we have database of variations, and we also have CleanVar. So SNP, as we will see in later classes, indexes small scale polymorphisms. So the ones that are 1 to 3 nucleotides or even up to maybe 10, 20 nucleotides. Database of variation indexes bigger size structural variants that are bigger than like 50 nucleotides, and CleanVar indexes information about clinically important variations. So these are three databases which have been devoted to organizing the data about polymorphisms. For gene expression, we have geo datasets and geo profiles. These are actually one database, but like two different search interfaces. So GEO is Gene Expression Omnibus, and we have geo datasets and geo profiles. For protein domains, we have Conserved Domains Database or CDD. And in the past, this is a screenshot from a few years ago. In the past, we had a homologen database, which indexed information about interspecies similarities or homologies. And we also had biosystems, which had information about protein-protein and molecular interactions. But now they are not included in the NCBI list of databases. I don't know the exact reason, but I guess they're probably working on it to upgrade them to a better database. So these are databases that we have in here. We have a central database which is named Gene. And as I mentioned last time, when we're looking for bioinformatic information for a gene, a segment of the DNA, a segment of the DNA which is actively transcribed and produces stable and functional transcripts. When we're looking for bioinformatic information for a gene, we usually identify the entry or the record in the gene database first, and then from there we get connected to the rest of the databases. So gene database has a central position, and today we're going to learn about it in more detail. So this is the gene database in here, and these are all in alphabetical order, so that's where the gene appears. So if you look up NCBI gene database in your search engine, or if you go and open up the database menu, you can select the gene database, and this is how it looks. So as you're seeing in here, in the help menu or in the information that's provided in here, NCBI Gene integrates the information from a wide range of species. So it's not only human, and it has nomenclature, it has genomic location, it has information about transcript variants, as well as information about the reference sequences. So what we're going to do today is to do some searches in the gene database to get familiar with how it works and to get familiar with different filters that it has. So here I started from tumor necrosis factor. It's an important gene for immunologists. It's a pro-inflammatory cytokine that has a central role in a lot of defense mechanisms as well as inflammatory diseases. So if I put in tumor necrosis factor and press search, these are the number of entries or records that I get. So almost 100,000 different records are retrieved by the search engine. So this is beyond what one expects, because even if you consider that we have TNF in different species, we don't have that many species whose TNF has been basically uh, indexed in the database. So when we look at the entries that have been retrieved, we see that many of them are related to TNF, but they are not the TNF itself. Like this first one, I have TNF receptor superfamily, um, member 1A, which is a TNF receptor. So it's not TNF, but it's related to a TNF. The second one is the TNF itself. So we see tumor necrosis factor and it's Homo sapiens tumor necrosis factor. The third one is another TNF receptor superfamily member 1A. By the way, this one was for mouse, but this is for human. Then we have TRAF2, which is TNF receptor associated factor 2. Again, it's related to TNF, but well, it's not TNF itself. And we have more stuff in here, and here we have mouse TNF. So if you look at the organisms in here, we have 922 entries which belong to Homo sapiens, even though we humans in our genome have one single TNF, well, two copies, so maternal, paternal, but one TNF locus in our haploid genome. But here we are seeing 922 different entries. And we see a big number for mouse and for rat and some other organisms. So what this tells us is that the search that we performed has not been a very specific search. And if you look at the search details, which is the part that's NCBI search engine named Entree, it's named Entree, sort of translates our search phrases into a sort of more machine readable version. We are seeing that it's searching tumor necrosis factor in all possible fields in each of these records. And that is why we're seeing a lot of things that are TNF related, but they're not TNF themselves. So if I want to do a more specific search, this is what I should write. So I should write TNF and open square bracket, then write gene name, close square bracket, then an operator which is and in here. And if I'm looking for human TNF, I would put the name human in here, and again open square bracket, organism, which is abbreviated as ORGN, and close the square bracket. So if I do it that way, I would get only one hit, which is tumor necrosis factor for Homo sapiens. So this is important. So let's open it up. The way to correctly sort of query a database, this is the technical term for searching a database, querying a database, is to identify or to specify which field in each record you're looking for your keyword in. So if I'm looking for human TNF, I would use these square brackets, and inside the square brackets, I should put the name of a previously sort of designed field for the database, a recognizable field. So that would become TNF square bracket, open square bracket, gene name, close square bracket. So this would tell the search engine that I'm looking only for entries whose own name is TNF, and I'm not looking for like TNF related factor or TNF receptor or things like them. And then we use operators, which are Boolean operators, and, or, and not, which have to be uppercase. So in this part, the search engine is sort of case sensitive. And then add the second part of the search. So if I'm looking for human TNF, I have to put human. But then I need to specify the field in which I'm looking for a human. And that field is organism. You can also put it ORGN. And then together they will become the search that we are seeing in here. So here we need to get familiar with the concept of a field. When you have a database, imagine that this sort of ellipse in here that I'm showing is a database. The database comprises records or entries. So it has many entries or many records, and each of these records contain information about some particular topic. And in the case of gene database, each record contains information about a particular gene or gene-related element. So if you open up each record, you see that that record has multiple parts, and each of those parts is called a field. and each field covers a certain type of information. So that's usually the way that databases are organized. They contain many entries or many records, and each record usually is composed of or comprises multiple fields. And you see this very often in many different databases. By the way, records in the database are not static, so it's not like they remain the same all the time. So sometimes new records will be added when new information is provided, and some of the older records might be discontinued or removed from the database. So this should be kept in mind, because later we will see in gene database and the rest of the databases that the default when we do the search are the current records, and the discontinued or replaced records are not shown. So by this information in mind, let's go back to here and to this slide and look at one example, one very familiar example of different fields in a record. Here I have screenshots from an ID card. That's an Iranian ID card, so you can see it in like birth certificates or ID cards or even driving licenses issued by the governments. So each one of these records, each one of these ID cards can be considered a record which contain information for a specific individual. But then it has multiple fields. So you have first name, you have last name, you have ID number, you have date of birth, you have place of birth, father's name, mother's name. So many different fields, and each record, depending on what type of information it's going to contain, might have different fields. Like an ID card might be slightly different from a driver's license, might be slightly different from passport. So imagine that you go to City Hall's registry and ask the person who is in charge there, give him a name like Reza, R-E-Z-A, which is a common Iranian name, and then you ask the person to retrieve all records that contain Reza. And what you have in mind like are people whose first name is Reza, but you haven't specified that. So what happens is that when the person retrieves the records, he will bring up all of the records that have Reza as the first name, but it's not limited only to that. Reza might appear in the last name as well, like Rezai and Rezazadeh. So all people who have Reza in their last name would also appear, and also people whose father's name are Reza would also be retrieved, because you haven't specified the field in which you're looking for that name. So this is exactly the same for online databases like NCBI databases. And here I want to make a little digression and explain this concept of field and how to combine fields and operators together to do searches with different levels of sensitivity and specificity. I want to use PubMed to do that, even though it's not part of our bioinformatics course to talk about PubMed or literature databases. Nonetheless, because we're sort of more familiar with PubMed and Medline, I thought it would be easier. It would make it easier to understand if we use some PubMed examples. So imagine that I'm looking for the role of lymphocytes in diabetes, and I put diabetes and lymphocytes in here. So I'm using that operator and there. In PubMed, even if you don't put it, it has an automatic system named automatic term mapping that would kind of translate your query into something more complicated. But here I've used it so that we can sort of keep it consistent with what we are going to do in the gene database. So if I look for diabetes and lymphocytes, I would be getting a lot of papers. And when we look at these papers, like the first one, if I look at it, it's neutrophil to lymphocyte ratio, NLR, a predictor of microvascular complications in type 2 diabetes mellitus. So this seems relevant. This has some information about the role of lymphocytes in diabetes. But if we scroll down, and I've scrolled down a little bit to the fourth paper that has been shown, before I take the screenshot. It's written early pregnancy HDL-related inflammatory indices and risk of preeclampsia. So this doesn't seem to be related to lymphocytes and diabetes. So how come this particular paper has been retrieved? Well, because I haven't specified where I want diabetes to be located and where I want lymphocytes to be located. It could be in the title, it could be in the abstract. As you're seeing, there is a lymphocyte in here. It could even be in mesh terms. So it would still be retrieved, but it's not directly related to the role of lymphocytes in diabetes. And I go to the next page, I see a similar story. It's like hemogram-derived inflammatory indices and metabolic renal biomarkers as predictors of complications in acute cholecystitis. So this doesn't seem to be related to diabetes and lymphocytes, but it's the search engine has found diabetes somewhere in the record. And here I have high density LDL related inflammation and reproductive lifespan. Again, this doesn't seem to be very related. So what this tells me is that I need to perform a more specific search or query the database, which is PubMed in here, in a more specific way. And the way that I can do it is to limit the keywords that I have in here or the search words that I have in here to a particular field. So I know that if a keyword appears in the title of an article, that article would very likely be related to the concept that I have in mind. So I limited it to title, square brackets, and then TI or title in the middle. And now if I look at the number of articles that I'm getting, it would be much less. I'm getting 249 articles. And when you look at the titles, you see that, well, they have lymphocytes and diabetes, and they're very likely related to what I have in mind. So this is a much more specific search, but it has a downside. When you increase specificity, you decrease sensitivity. So it's possible that I have many articles related to the role of lymphocytes in diabetes, but the word diabetes or lymphocytes do not appear in the title. So it's related like in terms of the subject and in terms of the concept, but the words have not appeared in here. So we've increased the specificity a lot at the cost of sensitivity. So what can I do? Can I make it sort of a moderate in terms of specificity and moderate in terms of sensitivity? So I can do it this way. I can use TIAB or title/abstract so that the word can be found either in title or abstract. And this sort of decreases my specificity but increases the specificity. And I get 4,000 articles in here. So if I want to look at it from the perspective of the two metrics of searches, which are, as I mentioned, sensitivity and specificity, if I search it like this without specifying the field, diabetes and lymphocytes, find it anywhere that you can find in any of the fields, I would be doing a very sensitive search. So it retrieves all of the articles that have these two words somewhere in the article. But a lot of things that I'm finding are not very specific or not very relevant to the concept that I have in mind, which is the role of lymphocytes in diabetes. So if I limit the search to the title of the articles for both of these search words, then I would be getting 249, which is a fraction of the initial number that I was getting. So the search would be very specific, but I would be losing sensitivity. So there might be articles which are important, but they don't have diabetes and lymphocytes in the title. And finally, I can come up with a sort of a moderate search strategy, either title or abstract, and then combine them like this, and that would give you moderate sensitivity and moderate specificity. So this is how you can use different search strategies to get to the results that you want, depending on whether you want to find all of the articles which have been published, or you want to focus on a very limited number of articles which are highly related to your search. So uh, I thought that would be useful before we move on to Gene. And actually, it would be nice to look at the relation between the so-called Boolean operators and not, and the so-called set operators. Although I guess you're familiar with that, but let's refresh our memories. Anyways, when we use and, this is a Boolean operator, and we have two sets, and imagine that these little red dots are the elements in the set. We are looking at the intersection between the two. So A and B, it means that these two elements which are common between two sets. If I use or, I'm looking at the union. So A or B, it means A and B together. And by the way, this is called inclusive or. So I'm looking for the members that are either in A or in B or both of them. So that would be okay if it's in both of them. We also have another OR which we don't use in database searches, at least not in NCBI database searches, which is XOR. And this is exclusive OR. So exclusive OR is looking for elements that are either in A or in B, but not in both of them. But the OR that we use in NCBI searches is basically inclusive OR. And finally, if we use A not B, that means sort of subtraction. So things which are in A but not in B. So keeping that in mind, I want to do a more complex search, to do a more advanced search by combining some of these operators. Imagine that I'm looking for breakthroughs in the field of diabetes, and I know that breakthroughs are usually published in journals like Science and Nature and Cell. So let's see what I get if I do the search like this: diabetes in title and science. And by writing this, I mean I'm looking for articles that have been published in Science. So what I'm getting in here is 83,000. But when I look at the articles, when I look at the journals, I see that these journals are not Science. Like this first one is Diabetes Care. The second one is, I think, Computer Informatics for Nursing or something like that. But they do have diabetes in the title. So what's wrong with this search? Well, the problem with this search is that the word science is a general term. So I haven't specified the field. If I want to do it correctly, I have to specify the field to JOUR or journal. And if I do it this way, I would be getting 185, and the journals are science and diabetes in here. So this is good. So far, so good. So imagine that I want to add the journal Nature. So Science and Nature, as well as some other journals like Cell, New England Journal of Medicine, Lancet, these are journals that major scientific breakthroughs are published in. So imagine that we're looking for papers that have been published in Science and Nature, or let's say Science or Nature. So if I write my search term like this, that would be diabetes and science and nature, I wouldn't be getting anything. No results were found, because this is looking at the intersection between science and nature. And that's not something that we have. We don't have papers being published at the same time, the same article in two journals. So the way that we have to do it is by using OR. So union of science and nature. So if I write a diabetes title and science or nature, again, the result that I'm getting is not what I expected. So I'm getting a huge number of articles, which I didn't expect because, well, I don't think that many articles have been published in these two journals about diabetes, or at least with diabetes in title. And when I look at the journals, I see Nature in here, but there are no diabetes in here. So why this has happened? It means that, well, the search phrase or the syntax that we have written in here is not working correctly. The problem is that when you start using more than one operator, the operators by themselves, they don't have any priorities. They're not like mathematical operators where multiplication and division are done prior to addition and subtraction. So and, or, and not per se, they don't have any priorities. So they start getting done from left to right, from left to right. So what happens is that we have three sets. One set is the set of articles that have diabetes in their title. Then we have a set of articles that have been published in the journal Science, and then we have the set of articles that have been published in the journal Nature. So what we have in mind is that we are looking for articles that have diabetes in title and they've been published in Science or Nature, or Nature. But the problem is that this part is sort of applied after the AND operator has finished its job. So the way that this works is that all articles that have diabetes in title are intersected, or their intersection is found with all the articles that have been published in Science. So the ones in here would be found. Then all of the articles in Nature will be added to them. So or in here, it means the union of the Nature journal with previous articles. And this is not what we intend to do. So the way to do this search is to use regular brackets or parentheses. So these are square brackets used to identify a field, and these are regular brackets or parentheses in order to prioritize one operator, one part of the search over the rest. So if I do it this way, the science journal or nature journal will be applied first. So a big union will be formed between science and nature journals, science and nature articles, and then the intersection will be calculated with the papers that have diabetes in their title. So I know this was conceptually simple, but this is important to be familiar with the correct use of parentheses. And if we don't use them, the way that the search is being to be applied is to start from the operator on the left hand side, moving all the way to the right. And actually I could have done it the other way. If I wanted to avoid using parentheses, I could have written science journal or nature journal first, so that the union is formed in here, and then and diabetes title. So that would have worked as well. But if you want to use parentheses, it would be easy to do it. So if I do it like this, I'm getting 413 results, and these are nature. And if you scroll down, we will see science articles, and then diabetes in here. So I use these PubMed examples in order to better clarify the use of operators as well as fields, and also when we want to combine different searches. So here we have a search phrase or a syntax, if you want to call it like that, which is using two fields, gene name and organism, one operator, which is and the only part which matters to be uppercase is and, the rest of it doesn't matter. So if you want to show it like Venn diagrams, that would be like this TNF gene name and human ORGN, and we have one single TNF in here.